Methods for isolation of microorganisms. Strict plate method, spread plate method and pour plate method. Strict plate method. It is a technique used to isolate a pure strain from single species of microorganism, often bacteria. Take a sterile nuclear agar petri plate between two oxidized burner. Sterile while look in burner for 15 to 20 seconds. Open bacterial culture tube between burner maintaining aseptic condition. Dip the vial look in culture medium and then streak the zigzag pattern with vial look in nutrient agar media. Sterile vial look again in burner for 25 to 30 seconds. Incubate petri dish 37 degrees Celsius for 24 hours. Observe the colony characters of isolated bacterial colonies on nutrient agar plate. Microbial culture, vial look, nutrient agar plate, Bunsen burner are the requirements. Here, as we can see, we are taking a sample from microbial culture and sticking it into a zigzag pattern between two Bunsen burner, maintaining well aseptic condition to avoid it from contamination of atmospheric microorganisms. Monostreaking, one flame, two flame or two sector are the types of streaking. It also includes 3 sector or 3 flame method, 4 sector or 4 flame method. Spread plate method. It is a technique to plate a liquid sample containing bacteria so that the bacteria are easy to count and isolate. Take a sterile nuclear agar media petri plate. Take sterile pipette and take a notified amount of bacterial culture with pipette and put in the petri dish between burners. Take a spreader and dip into 70% alcohol and spread culture moving spreader and petri dish in the circular direction till surface of an agar media becomes rough. And then incubate the petri dish 37 degrees Celsius for 24 hours. Observe the colony characters of isolated bacterial colonies on nutrient agar. Pipette, spreader, microbial culture, Bunsen burner. As we can see, we are taking some sample from microbial culture and putting it into a nutrient agar. And then we are spreading it between two Bunsen burners to avoid it from contamination of microbial. Here the result of spread plate method. Pour plate method. This technique is used to isolate microbial colonies by serial dilution and then counting the colony forming units. The procedure includes perform serial dilutions. It can vary. Take sterile molten agar nutrient plate. Take a pipette and take some notified amount of bacterial culture and put into a molten nutrient agar and mix it smoothly between two burners. And then leave it for 4 to 5 minutes for stability. Incubate 37 degrees Celsius. Observe the colony characters. Molten agar, microbial sample, Bunsen burner, petri plates. As we can see, we are taking sample from microbial culture into a molten agar between two oxidizing burner, maintaining well aseptic conditions to avoid the contamination. Here we are pouring it into a sterile petri dish. That is why it is called pour plate method between two oxidizing burner. As we know the reason. Here are the results of pour plate method. Thank you.